friends, welcome to my YouTube channel Sam Biology Class. Today we will discuss about different types of microbial culture media. But before that, will you please consider subscribing my YouTube channel? Your subscription and viewership are often a lot to me. Microbial culture media are nutrient preparations used to grow, cultivate and identify different types of microorganisms. A culture medium should contain a suitable amount of water, a source of carbon, nitrogen, growth factors and essential minerals. Based on consistency, culture media divided into three types. Number one, solid media, which contains agar as the solidifying agent. In such a medium, the amount of agar accounts for 1.5 to 2%. Examples are nutrient agar, blood agar, and McConkey agar. Solid culture media are commonly used to isolate pure microbial colonies for antibiotic testing, to study colony characteristics and for long-term storage. Number two, semi-solid media in which the amount of agar accounts for 0.5 or less than 0.5%. Because of the presence of a very small amount of agar, such type of media has a jelly-like consistency. Examples are Stewart's and Amis media, Hoff and Hepson's fermentation medium, and mannitol motility media. Semi-solid media are commonly used to cultivate microaerophiles and to study bacterial motility. Number 3. Liquid culture media, which is also known as broth. It do not contain agar. Examples are nutrient broth and tryptic soy broth. Liquid media are used for large-scale industrial fermentation, for biochemical testing, for stock culture maintenance, and for research diagnostics. Based on whether the exact chemical composition of the media is known or not, culture media are divided into two types. Number one, undefined media which has a complex chemical composition. Its exact chemical composition is not known. It is prepared by mixing complex organic nutrients such as blood, serum or egg yolk. Examples are McConkey agar and potato dextrose agar. Number two, defined media, which is also known as synthetic media. Its exact chemical composition is known. It is prepared by taking recommended measured amount of various nutrients and mixing it with suitable amount of water. So exact chemical composition and the proportion of various nutrients in a defined medium is known. One example is Zapedox media. Based on functional use, culture media are divided into several types. Number one, basal media, which can be solid or liquid. It contains basic nutrients such as glucose, nitrate or ammonium salts, growth factors and minerals. Basal media is commonly used for the growth and cultivation of a wide variety of non-fastidious microorganisms. It ensures basic cell survival and is used for routine subculturing, isolation and maintenance. Examples are nutrient agar and pectin water, which are used for the growth of Staphylococcus and Enterobacteria. Number two, enriched media. It is a solid media containing complex organic nutrients such as blood, serum, or egg yolk. Enriched media is widely used for the growth and cultivation of fastidious microorganisms. One example is blood agar, which is an enriched differential medium. It supports the growth of Streptococcus, Neisseria, and Haemophilus. It is also used to classify bacteria based on their ability to destroy red blood cells. Second example is chocolate agar, which is used for the isolation of Haemophilus species and sexually transmitted bacteria such as Neisseria gonorrhoeae. Third example is Loeffler's serum medium, which is used for the growth and cultivation of Corynebacterium diphtheria. Number 3. Enrichment media. It is a liquid media commonly used to increase the population of a particular microorganism from a mixed sample. It contains specific chemicals, antibiotics, or dyes that promotes the growth of a particular microorganism while inhibiting others' growth. Such medium can also use an altered pH that promotes the growth of a particular microorganisms. One example is selenite broth, which is used to recover Salmonella species from fecal sample. Another example is alkaline peptone water. It is used for the growth of Vibrio species from different samples by taking the advantage of its high pH. Fourth one is differential media, which is also known as indicator media. Such type of media are commonly used to distinguish between different microorganisms growing on the same nutrient plate. This distinction is based on the biochemical and metabolic characteristics of the microorganisms, such as their ability to ferment certain sugars. Differential media contains specific chemicals known as indicators, such as phenol red, neutral red, or methylene blue. When grown in such type of media, microorganisms having different metabolic pathways consume specific substrate present in the medium and break down into byproducts that alter the pH of the media. This results in a distinct visible change in colony color or in the surrounding medium. This helps us to identify specific bacterial colonies. One example is mannitol salt agar, which contains high salt concentration that prevents the growth of microorganisms which are non-halotolerant. This medium contains phenol red as the indicator. Mannitol salt agar is particularly useful for the isolation of Staphylococcus aureus. When grown in such type of media, Staphylococcus aureus ferment mannitol into acidic byproducts that alter the pH of the media. As a result, the color of the Staphylococcus aureus colonies changes from red to yellow. Most other Staphylococcus species fails to grow. Number 2, blood agar, which is used to distinguish between hemolytic and non-hemolytic microorganisms. Blood agar do not contain an indicator, but the blood itself acts as a biological indicator. Number 3, McConkey agar. This medium contains crystal violet and bile salts which prevent the growth of gram-positive bacteria while allowing the non-fastidious gram-negative rods to thrive. This medium contains neutral red as the indicator. This medium is used to distinguish between lactose-utilizing and non-lactose-utilizing microbial colonies. When grown in such type of media, lactose-utilizing microorganisms consume lactose and break down into acidic byproducts that make the target bacterial colonies red. On the other hand, those microorganisms fail to utilize lactose. Their colonies remain colorless or transparent. Number 5. Transport media, which is useful for the transport of different types of microbial cultures. Transport media prevent overgrowth, maintains viability and prevent the bacterial cells from drying. Examples are carry blades, tea media and Pike's media, which are used to transport streptococci culture. Number 6. Storage media, which is used for storage of different types of microbial cultures. It increases the longevity of the microbial cells and is useful for storage for a longer time period. Examples are cooked meat broth and NaX saline. So friends, this is all about different types of culture media. For more updates, subscribe my YouTube channel, Sam Biology Class.